Okay, so for today's online lab, what we're going to explore is the Enteropluri test for identification, as well as testing to analyze or look at the motility of different bacteria. And both of these testing abilities are really valuable when you're dealing with unknown bacteria, especially gram-negatives in the case of the Enteropluri test, and you want to gain more information about these bacteria. So, like I just said, the focus is going to be with regard to unknown bacteria and basically identifying and characterizing bacteria. Now, when you think of dealing with, bacteria that exist that you haven't really identified or found yet, there are four main reasons that we end up looking to identify and characterize microbes. Now, the first one makes a lot of sense, especially nowadays, and that is primarily the medical aspect. So it's really important for us to be able to identify and characterize microbes because a lot of them are pathogenic and deal with infectious disease. Okay. So you really want to gain understanding and be able to identify or culture patients when they're dealing with different infections. Now, not all microbes are going to be looked at in the medical sense. We also end up needing to identify and characterize bacteria for industry as well, because as we've talked about in the lecture, there are a lot of different enzymes and proteins and metabolic end products that we can really use from different bacteria. So some of the ones that we've mentioned a lot are things like enzymes that are used in labs, such as Taq polymerase for PCR, but also things that you're very familiar with, like antibiotics. We've already mentioned in lecture that antibiotics came from fungi and bacteria. So by identifying and characterizing different microbes, we were able to extract and mass produce various of their very valuable products. It's also valuable to us in the food industry to be able to identify and characterize bacteria because a lot of these bacteria make things or perform certain processes such as fermentation that can really benefit us. Okay, so in the examples, it's funny when I say benefit us, the first thing is alcohol, but also things like dairy and yogurts, breads, all of that production. If you think of yeast fermentation, Saccharomyces cerevisiae being used to produce beer and breads, if you think of all of the probiotics and things like lactobacillus that you find in yogurt products, things with cheeses as well. So any of that stuff we were able to produce and make better thanks to identifying the microbes that could help us with it. The last of the reasons that we have for identifying and characterizing bacteria is a crazy looking term called taxonomy. Basically, this is, looking at evolutionary comparisons, making things like phylogenetic trees. Okay, all of this is, fancy terminology for saying making the trees, the family trees, of which bacteria are more closely related and which ones are more distantly related. And you could ask yourself, well, why does it matter, which bacteria are bacteria cousins? Well, it matters because think about if bacteria A, you isolated and found that it makes a very valuable, helpful antibiotic for you, well, maybe its close relative, bacteria B, may also produce an antibiotic that's similar or maybe even better. So it's very valuable to us in all of the other categories that we just talked about when you really learn about which bacteria are closely related and which ones are more distantly related. It even helps with things like treating the bacteria because, if they're more closely related, odds are the same drugs or therapeutics will work against them versus the ones that are very distantly related. Okay, so keep all of these in mind with regards to why you would want to identify or characterize unknown bacteria. Now, when we talk about identifying unknown bacteria, the main term that's going to keep coming up with that is biochemical fingerprinting. When you ask yourself how you're going to identify an unknown culture or unknown bacteria, well, a lot of the times we're able to do that based upon that microbe's enzymatic activities. Okay, so you've actually seen this already in the lab. For instance, when you saw MSA plates. When Staph aureus or Staph saprophyticus were present, ask yourself, what happened? Well, the red plate turned yellow because they were capable of enzymatic activities of breaking down the mannitol, right? And you were able to visualize that with differential media. So that's what we mean by biochemical fingerprints. That yellow change in the media is the fingerprint of those organisms. Now, when we say enzymatic activities and looking for end products, we're dealing with metabolism again, and I want you guys to circle star highlight metabolism. You know, we learned about this in lecture, we learned about it in lab. Make sure you know that metabolism means all of the chemical reactions in a cell or body. This includes anabolic and catabolic. Now remember, the way you can distinguish those two is picture a cat scratching apart a sofa because it's breaking it down. So what that bullet point means is that metabolism includes building up and breaking things down. Now it's important that you include both of those because so many students, when I ask them what metabolism is, they only think of your body breaking down food. And that is only half the answer. It is also your body building up and making all of the biomolecules that we've talked about this semester. Now, in terms of biochemical fingerprinting, the way that we're able to identify and classify microbes is by the metabolism or metabolic end products. Okay, we're able to assay or use differential media to try and identify the unknown bacteria based on which enzymes, which reactions that bacteria performs. So one of the tests that you can use to identify an unknown bacteria from a culture is the Enteropluri test. So the Enteropluri test is this tube that you see in the corner of the slide, and it has 12 different sectors, each with a different media, so different agar. Kind of picture in lab when you've worked with agar plates, picture 12 of those agar plates lined up in one tube. Now, interestingly, even though there are 12 sections, it actually does 15 individual biochemical tests for you because some of these sections do more than one test for you. So for instance, the first one, it has that wax on top of it so that you can see if in addition to fermentation, you're getting gas production. Okay, and some of the other sections you'll add additional chemicals to look for further changes. So please remember that the Enteropluri test has 12 sections but can do 15 tests for you, 15 metabolic tests. The other thing I want you to remember, circle, star, highlight the fact that this Enteropluri test, where you take a colony of bacteria and you drag it, you place it onto the needle and you pull that needle through to get the bacteria through all of those pieces of agar, this test can only identify gram-negative bacteria. Okay, so we put it as Enterobacteriaceae and other gram-negatives, and we also mention oxidase negative because that's an enzyme involved in the electron transport chain. With that extra information, it's okay if you don't remember the Enterobacteriaceae term, if you don't remember oxidase negative. What I want you to remember is this will only work to identify gram-negatives, okay? Because ultimately at the end of this, you will look at all 15 of those test results. you will come up with a 5-digit code, and you'll look in a booklet that has the different possible identities based on that number you come up with. Now, you will get a number that matches nothing if you try to test something that is a gram-positive. So for instance, if I ever asked you, can you use the Enteropluri test to identify staph or strep? So for instance, previously we looked to identify Staph aureus or Strep pyogenes. Well, can you use the Enteropluri test to identify either of those? The answer would be no, because both of those are gram-positive, okay? This test will only identify gram-negatives. So I want you to be able to answer that question if I asked you, could you use Enteropluri for identifying staph or strep? If not, why not? Explain that they are gram-positives, and this only works to identify gram-negatives. Now I threw in this slide just so that you can see the 15 different tests that this does. So basically, like we said before, there are 12 sections of the tube, 15 different tests, and all of them are metabolic processes that we're testing for. We want to see, does that unknown bacteria ferment glucose? If it does it produce gas? Does it break down the amino acids lysine or ornithine? Does it produce iron sulfates and hydrogen sulfides? Does it break down tryptophan and produce indole? Does it ferment any of the 4 sugars that we have listed as inositol, lactose, arabinose, sorbitol? We look for the VP tests, where you're testing one of the intermediates of glucose fermentation. There's dulcitol fermentation, breaking down phenylalanine. There's urea hydrolysis, or using citrate as a carbon source, okay? You do not have to memorize these 15. You will always see them listed out. I just wanted you to be able to recognize, that these 15 processes that you're checking for are metabolic reactions, but you're not memorizing the names. You will always be given them. They're on the side of the tube. Now, in addition to the Enteropluri test, I also want to introduce in this lab motility, because characterizing motility of an unknown bacteria can really help you narrow down which bacteria that can be. So first off, you should be able to answer, what exactly is motility? Well, that's the ability for a bacteria or any organism to move on its own. Now, bacteria will use 3 main mechanisms for motility, and you've seen a lot of them in lecture already. So you have flagella, axial filaments, or gliding motility. And we're just going to briefly review what each of these means. So the first way that a bacteria may have motility is by having flagella. And I want you to know the different terms, because different bacteria will have different flagella if they have any flagella at all. Okay, keep in mind, only certain bacteria will have motility, only certain bacteria will have flagella. So if a bacteria has flagella, you'll see the word trichus. So you'll see at the end of the words trichus, as soon as you see that, that tells you you're talking about flagella. If a bacteria is monotrichous, well, mono means 1. So the bacterium will only have a single flagella. So for instance, even some cells in humans can be monotrichous. For instance, a sperm cell is monotrichous. Then you have the option for lophotrichous, and I think LATSO for lopho. Lophotrichous is basically a bacterium with a ponytail of flagella. So it will have a tuft on one side of its cell. Okay, so that's lophotrichous, a ponytail look to it. Amphitrichous means that the bacteria will have 2 flagella, 1 on each end. Okay, so when you hear amphitrichus, that's 2 flagellum, 1 on each end. The last one is peritrichus, and under peritrichus, I want you to write perimeter, because I'm sure you've all heard the term perimeter, such as walking around the perimeter of your house. So perimeter means all around, right? The outer edges. So peritrichus means that the bacterium has flagellum all around its entire cell, okay? Around the entire perimeter of the cell. Now, no matter which type of flagella structure or organization the bacteria has, it will basically whip those little flagella around and propel or move in the direction that the flagella force provides. Some bacteria instead will have axial filaments, and we've mentioned this before in lecture as well. When you hear axial filaments, you have to think of one of my favorite infections to mention, which is right here in the corner, syphilis, okay? Treponema causing syphilis has axial filaments, and thanks to that, syphilis can embed in tissues and become a nasty infection. Now, what axial filaments means, remember, one of the other terms that you hear it called is endoflagella. Okay, endoflagella because axial filaments are under the surface of the bacteria. So it's almost like it's internal. Okay, so it's axial filaments you find wrapped around spirochetes or underneath their outer sheath. Okay, so notice here the axial filaments are underneath the outer sheath. Okay, and they use those axial filaments to corkscrew or spiral around. So whenever you hear axial filaments, please remember syphilis and spirochetes in general. Okay, make sure to write that down. Spirochetes use axial filaments, including the one that causes syphilis. The last category of motility is gliding motility, and researchers don't fully understand the proteins and mechanisms involved in this one, but this is basically if you've ever seen kids playing with a slip and slide, okay, so, or if you've ever seen anyone spraying down, any kind of slide, water slide, and sliding down on it, or even a slug you can think of for this one, because in this gliding motility, the bacterium will basically secrete slime, and it will make a slime trail for itself to then slide across and move. Okay, so when you hear gliding motility, think a layer of slime secreted to slide across. Now you may ask yourself, how can we test to figure out whether or not our unknown bacterium is motile? So what we do is called a soft agar stab. So basically we make a test tube with agar in it, just like your agar plates that you've worked with this semester, but instead of being that dense, we put less of the solidifying agent in there. So it'll be a little softer, and what you do is you take an inoculation needle, so a straight metal needle, you dip that into bacteria, and then you gently and carefully stab that through the center of the agar and gently pull directly up, okay, without shaking, without moving. So you stab once and pull back exactly how you had stabbed into that media. And then what you think of now is you just placed that bacteria in a straight line in the center of that agar. If it's motile, what will it do? It will move around. It'll move away from that line. If it's not motile, then it will stay in place. And I'm going to show you a picture of what we mean in the next slide. But first, I just want to mention that sometimes, in addition to using regular standard agar, like the TSA that we use, sometimes they also add things such as TTC, a chemical that will allow red pigment, or they use agar such as SIM or MIU, which has multiple differential aspects. So in addition to being able to see if your bacterium, your unknown bacteria, can move on its own, you'll also be able to see some of its metabolic processes, which again helps you further characterize or identify the bacteria you're dealing with. So things such as sulfur reduction, indole, urea hydrolysis, the things that Enteropluri would test as well. So here's a visual so that you can see what I was talking about. Like I said, sometimes they'll put TTC in the agar so that any bacterial growth will come out red. Sometimes instead there will not be anything, no TTC, and so the bacteria will look beige. But either way, the premise is the same. You start out with a clear, uninoculated agar. You then place the bacteria in a single stab down the center. Now, what do you notice about this line? The bacteria stayed exactly where you stabbed it, where you placed it into that agar, because this bacterium is non-motile. Okay, if you ever look at a result like this one or like this one, it would tell you if the bacteria stayed in on a line, if you can see it as a defined line, then that bacteria is non-motile. It does not have flagella or any other way to move. It stays put. If, however, suddenly the whole tube turned red in that case, or in this case, the whole tube becomes cloudy, okay? You can't see a single line. Instead, the whole tube became cloudy. Why did it become cloudy? That bacteria is motile. It moved away from the single line. And if it moves away from these single lines, the whole tube ends up having growth, okay? So make sure you're very clear on what it looks like if a bacterium is non-motile, such as right here and right here, or if it's motile, which are the end tubes on the right of each picture. So you see cloudiness, you do not see a single line, okay? Now, if you were thinking of the bacteria itself, when you see something like this, where it turned cloudy, well, that would be a bacteria where you see flagella on it or where you see axial filaments on it, okay? If you see something that's just, round or rod-shaped and has nothing protruding out of it, then odds are it would stay in a straight line, unable to move on its own. So now we're just going to go over some of the protocol information. So if you're working with an Enteropluri tube, you start out with the capped tube and you have a plate of unknown bacteria swabbed on there, so lots of culture. And what you do is you clamshell open the tube, and when you uncap your Enteropluri tube, you see that there's a metal needle, inoculation needle, running through the whole tube. So what you do is you scoop up some of the bacteria onto the tip of the needle and pull that needle through all of the sections. And that will place some of the bacteria in each of the 12 agar plates within that within that tube. Okay? And then you'll look to see what changes occur after incubating it. So like I said, you would incubate it. Usually we incubate it for 24 to 48 hours, a lot of times at 37 degrees, which the bacteria like. And then you'll look at all 12 of those sections and look for any color changes. You'll then perform 2 extra tests, which are the VP section and the indole section. And what you do is you cut open the plastic wrapping above each of those sections, and you put extra drops of chemicals into each one. For indole, once you put the droplets of Kovac solution into the indole section, if that microbe is indole positive, you'll see pink suddenly appear. For VP testing, you'll open up the VP section, you add some droplets of alpha naphthol and KOH, and if that section is positive, you'll see red, but that reaction takes a bit longer, so it takes 20 minutes. Now, ultimately, the way we analyze these enteropluri tubes is we think of what the tube looks like before inoculation, all of the initial colors, and then we look for any changes in color or the lifting of any wax showing gas production. Here I mapped out all 15 of the tests, and when you start off with an enteropluri tube, these are all the original colors, okay? If you see a change in color, you then have a positive result for that test. So for instance, if your glucose section, which started out as red, is suddenly yellow, you would mark that as being positive for glucose. If you see lifting of the wax, or what I call a bubble formation, that would mean your gas section is positive. Purple in any of these two sections will tell you positive. Okay, so all of these results are the positives, and here are the negatives. This will help you a lot when you're trying to analyze your results. Now keep in mind, I know it looks scary thinking of analyzing so many different tests at once, but a lot of these are sugar fermentation, such as glucose, inositol, lactose, arabinose, sorbitol, and dulcitol. And you already know from MSA plates what color to expect if fermentation is occurring, which is yellow. So you've already kind of encountered some of this material before. Now, when you go to analyze an enteropluri tube, you always want all 15 of the tests lined up in order. Okay, we never move around the order. We keep them always in the same order that you find them on the tube. You then go one by one and ask yourself if the result that you got was positive or if it was negative. So for instance, if you're looking at your enteropluri tube and you look at glucose and you see yellow, you would check off positive. If, however, you looked at the glucose section and it was still red, you would check off negative. Okay? Now, when you analyze an enteropluri tube, there are two things that you'll want to do first. One is divide up your 15 sections into 5 groups of 3. Okay? So whenever you get a chart to analyze for enteropluri, one of the first things you'll want to do is, so the first thing you'll want to do is draw a line for yourself after every 3 tests to remind yourself what groups you're dealing with. Because when you go to make your 5-digit code, you're basically going to have 5 different groups of 3. Okay? And we're going to see this in the next slide where I help analyze a result with you. Okay, so analyzing enteropluri. When you're going to analyze an enteropluri, remember the first thing I said you should do is separate your groups on your chart. So go 1, 2, 3, glucose, gas, lysine, I make a line. Ornithine, H2S, indole, that's another 3, I make a line. And after every group of 3, I'm going to make a line. Then what I'll do is I'll go test by test and ask myself, is it positive or is it negative? So the first test we're looking at is glucose. The result says it turns yellow, which is a positive result. For gas, we saw a bubble, which is lifted overlay, or wax. So that is also positive when you see a bubble. Lysine stayed yellow, so that's negative. Again, you could look at the chart that I had a few slides before this when you're analyzing. You don't have to memorize what is positive, what's negative. Ornithine is purple. Sorry, that's positive. H2S and indole both remained regular beige colors. So, sorry, the screen keeps messing up. Negative and negative. Then you have adonitol staying red, which means no fermentation, that's negative. Lactose and arabinose, yellow and yellow. If we looked at the chart on the previous pages, that would be positive. Sorbitol red is a negative result. VP red is a positive result. Dulcitol and PA both green, those are both negative results. Urea pink, which is one of my favorites, is a positive result, and citrate, another favorite of mine, turns a beautiful blue if it's positive. Okay, so now we have listed out by checking on the chart on the previous slides what's positive, what's negative. Okay, so the first thing you do is you draw the lines to separate your groups. The second thing you do is you check off what's positive or negative. Now it's time to start calculating your positivity numbers. So what you do is any value that's negative, you go in and you draw a 0 as the positivity value for anything that you put a negative for. Okay, all the negatives get a 0. Okay, now it's time to convert all of the positives into positivity numbers. The way that works is any first test, so first test in a group, if it's positive, it becomes a 4. Any second test in a group, if it's positive, it becomes a 2. And any third test in a group, if it's positive, becomes a 1. Any negative, we already said, is a 0. So any negative is a 0. Okay? So glucose, that is the first test in that group of 3. So when that is positive, 4. Sorry, the slide was messing up when I was trying to draw on it. So that becomes a 4. Gas, which is positive, is the second test in a group, so that would be a 2. Lysine, we already said, is negative, so that's 0. Ornithine, that's the first test in that group, so that gets a 4. We have a bunch of negatives, which are 0. Lactose is the second test in a group. Positive, so it's a 2. Arabinose is the third test in a group. Positive, so that gets a 1. Sorbitol, we already said, was negative, so that's 0. VP is a second test, so that gets a 2. Dulcitol and PA, negative, so that's easy, they're both 0. Urea, what is that? That's a second test in a group, so that gets a 2. Citrate is a third test in a group, so that gets a 1. So remind yourself 4, 2, 1, okay? Any first test, which would be glucose, ornithine, adonitol, sorbitol, or PA, anytime those are positive, they would get a 4. Any second test, which would be gas, H2S, lactose, VP, or urea, those would get a 2 if they're positive. And any third test, such as lysine, indole, arabinose, dulcitol, or citrate, they get a 1. Okay, so now you have all of your positivity numbers. Now the last thing you have to do is come up with your group positivity numbers. In order to do that, you go group by group and you add together your 3 individual numbers. So 4 plus 2 plus 0 would be 6. Okay, 4 plus 0 plus 0 would be 4. 0 plus 2 plus 1, that would be a 3. 0 plus 2 plus 0, that would be a 2. Sorry, I should have kept showing you the brackets. and 0 plus 2 plus 1 would be a 3. So now your 5-digit code, you would just rewrite all of these in order, would be 6, 4, 3, 2, 3. That would be your final 5-digit code, and you would look that up in the Enteropluri manual, which I'm going to have as an extra PDF in the folder on your Blackboard, and you would look up to see which bacteria matches that number, 64323. 64323, you would then write both of these values here, both of those possible identities, which are Enterobacter georgiovi and Enterobacter cloacae. Okay, so you would have narrowed it down to 2 possible identities, and you would write down both, and then further testing could be done. Now notice that sometimes there's only one bacteria listed for a number. Sometimes there's multiple. If there's only one, you would write the single one. If there's multiple, you would write all of the ones you see for that particular number. And again, you're going based on your 5-digit code, which you looked up there, and find the matching bacteria. Don't worry about what's written here. That's basically telling you that sometimes certain tests will kind of be giving atypical results, and sometimes that's why you see differences in the possible identities, okay? But you don't have to worry about that. Now for the results, you're going to end up being responsible for identifying and analyzing your own Enteropluri tube results here. So notice that first I give you the uninoculated result. Okay, so that's the tube before you put any bacteria. Then you get your unknown X bacteria changed the results to look like that. So after incubation, you notice all of those color changes. Now it's very important to pay attention in the results set to what I wrote up top, because some of the results are going to be a little difficult to see. And so with that, I want to point out, so for instance, gas. In this picture, it's tough to see whether or not there's a bubble. So I pointed out here that there is a gas bubble. For indole and VP, because those are what you add afterwards, it's a little tough to actually see them as well. So basically I wrote up top that, you know, there was no change, telling you that it's negative. Okay, so then you would then go test by test, go for glucose, go for gas, go for lysine, for each one asking yourself, do you see a positive result or a negative result? So for instance, glucose is yellow. Does that tell you it's positive or negative? And you would do that for all 15 tests, and you should then be able to come up with their positivity numbers and their group numbers, and then look up and identify unknown X. You will then be able to do the same thing for unknown Y. Okay, so unknown Y, the final results are right here. I also put some information up top again. So if you're having trouble in the picture seeing, notice that the gas bubble is present. Notice that there is no change of VP, but there is red color for indole, or pink color, sorry, for indole. I should have written pink. And also note that over here for the PA results, so let me find it on here. This section here is dulcitol and PA. When you end up with a positive for PA, it makes it difficult sometimes, especially in pictures, for you to see whether the dulcitol was negative or positive. So I wrote up top to let you know that dulcitol is negative. Now, when you're analyzing your results, if you have any trouble, any confusion, just please send me a remind message and I will help guide you through it. You then have results set 3, which instead of enteropluri is now focusing on motility. So with this, it's critical thinking. So I gave you a picture of what staph looks like, which you're all very familiar with, those grape-like clusters, and also what E. coli looks like. And notice the difference between the two. You then have to figure out in the post-lab your analysis of unknown N and unknown K. Notice the difference between the N media versus the K media. That tells you information about their motility, which then should help you look at your staph and E. coli picture and say, hey, if a particular unknown is motile, which one's it more likely to be, staph or E. coli, looking at the pictures? If it's non-motile, which one's it more likely to be? Okay, so not only can you interpret the results of the tube and figure out what the unknown, is characterized, you can then narrow down the identity, especially if you know, okay, one is staph, one's E. coli, tell me which one's which. You should be able to do that. And if you're having trouble seeing any of the pictures or kind of understanding any of it, again, just contact me and remind, let me know, and I will help you out. Okay? Thank you, and have a great day.